Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator here at Simulations Plus. I'll be hosting today's behind the scenes, making sure we don't have any technical difficulties. In this webinar, our panel of experts will discuss the evaluation and effectiveness of intranasal reversal agents using FDA's translational model of opioid overdose. We take your privacy right seriously. Before we begin, we'd like to go over some quick housekeeping. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. This panel will address all questions after the presentation. Dr. Amparo de la Peña, our Vice President of Pharmacometric Services, will serve as our moderator today. Go ahead, Amparo.
Thank you, Jasmine. So I'll start by presenting our presenter, who is Dr. Daniel Gonzalez. He is a clinical pharmacologist and pharmacometrician with expertise in applying mathematical modeling and simulation techniques to characterize the PK and PD of drugs and guide drug dosage optimization. Dr. Gonzalez received his PharmD and PhD from the University of Florida College of Pharmacy in 2008 and 2012, respectively. He then completed a postdoctoral fellowship through the UNC Duke Collaborative Clinical Pharmacology T32 postdoctoral training program. Dr. Gonzalez has published more than 115 peer-reviewed publications and more than 40 abstracts. Now I will introduce our panelists, which will be discussing the information presented after Dr. Gonzalez's presentation. So first, Dr. Jiwa Li is a pharmacokineticist at the Division of Applied Regulatory Science at the Office of Clinical Pharmacology in the Office of Translational Sciences at the Center for Drug Evaluation and Research at FDA. Dr. Li has multidisciplinary training and a strong track record of leading basic and applied research programs in diverse fields of genomics, computational biology, systems pharmacology, and mechanism-based as well as AI-based modeling. He has been leading the initiatives for CIPA, the Comprehensive In Vitro Proarrhythmia Assay, a large-scale international collaboration to develop a new regulatory paradigm for cardiac safety. This led to the revision of the International Council for Harmonization regulatory guidelines for cardiac safety, during which he served as one of the experts representing FDA. He also led the development of translational models to evaluate the effects of opioids and opioid antagonists, which played a critical role in regulatory decision-making processes, such as the approval of novel opioid reversal agents and the discussion of public health and other product-related policies. Dr. Li has received several awards from the federal government, including the FDA Scientific Achievement Award in 2023. Dr. Albert Dahan is Emeritus Professor at Leiden University. For more than 30 years, he chaired the Anesthesia and Pain Research Unit, a nonprofit organization that performs outcomes research in anesthesia and pain medicine with special focus on pain, opioids, opioid adverse effects, and their reversal and PK/PD modeling. Since the first of this year, he is the CEO of the MediID Consultancy Group, the natural successor to the Anesthesia and Pain Research Unit located in Amsterdam, the Netherlands. The center is working in pain medicine, analgesic efficacy and adverse effects, and their reversal and PK/PD modeling in close collaboration with various commercial and non-commercial partners. His current work is focused on reversal of opioid-induced respiratory and cardiac arrest with various naloxone formulations and agnostic respiratory stimulants, as well as treatment of OUD, ketamine, and acute and chronic pain. Dr. Mark Gold is one of the top 15 impactful addiction scientists, an inventor, pioneering translational researcher, eminent scholar, distinguished professor and chairman, and emeritus eminent scholar, whose career in translational neuroscience began in 1972. Dr. Mark Gold is considered an addiction research pioneer because of his groundbreaking contributions to the scientific understanding of addiction as a brain disease and its treatments. His most significant discoveries and contributions include, first, development of the dopamine hypothesis, The work of Dr. Gold and his team was instrumental in helping establish the dopamine hypothesis of addiction, which posits that addictive drugs stimulate the brain's reward system by releasing dopamine, leading to reinforcement of drug-seeking behaviors. Secondly, the discovery of the role of the endorphin system. His research into opioid addiction significantly advanced knowledge about how opiates interact with the brain's endogenous endorphin system, including the biological mechanisms of opioid dependence and withdrawal, helping to guide new treatment approaches. Third, the development of addiction treatment approaches. Dr. Gold has been a leader in advancing medical treatments for addiction, from opioid antagonists to treatment strategies for other substances, such as cocaine and alcohol. Fourth, the work on food on behavioral addictions. Dr. Gold established links between addictive substances and compulsive behaviors like overeating and gambling. His work helped frame the concept of food addiction, especially to highly processed, high-fat, and high-sugar foods. And last but not least, his public health advocacy. He has been active in translating addiction science into public health policies, advocating for improved treatment access, education on addiction, and recognition of addiction as a chronic relapsing disease. And then we have a last panelist that we're waiting for right now, who's Dr. Boyer. Dr. Edward Boyer is currently professor of emergency medicine at the Ohio State University Wexner Medical Center, an awesome place to work where people are very friendly and caring. Dr. Boyer graduated from Columbia University Medical School in 1995, did his internship and residency in 96 and 99 at the University of Pennsylvania, and his fellowship at Boston Children's Hospital in 2001. He is board certified by the American Board of Emergency Medicine and Toxicology. As an emergency medicine physician specializing in medical toxicology, he most often treats patients suffering from overdoses caused by either an illicit substance or from taking excessive amounts of medications. He also cares for patients who have been poisoned or are dealing with adverse drug effects. He is passionate about healing these patients to the best of his ability and considers it an honor to know his body of work has helped to refine how medical professionals manage opioid overdoses in both adults and children. Dr. Boyer's research is focused on how to utilize advanced technologies at the intersection of substance abuse, HIV, and human behavior. Training the next generation of physicians has been a highlight of his career, and he's very proud of his mentees who have gone on to successful careers of their own as hospital leaders, medical school deans, and department chairs. In his free time, he enjoys beekeeping and knows that as a scientist, this practice really tests and hones his powers of observation. So without further ado, Dr. Gonzalez, the podium is yours.
Thank you very much. Thank you, Dr. De La Peña, for the introductions, and thank you all for joining today's webinar. So the objectives of today's webinar are to provide an overview of the clinical need for opioid reversal agents, to inform the audience on the current available treatments, to discuss FDA's translational model with a focus on the updates described in the Lafont et al. 2024 publication, to compare the cardiac arrest percentages of predictions for both intranasal reversal agents, naloxone and nalmefene, and to address scientific questions from the audience by an esteemed panel of experts in the field. So in terms of conflict of interest disclosures, a validated translational model which evaluated the opioid reversal agent naloxone was published by Mann et al. In 2022 and Strauss et al. 2024. Lafont et al. In their 2024 publication leveraged the translational model to perform additional simulations to evaluate the effectiveness of intranasal naloxone and intranasal nalmefene. Intranasal nalmefene was developed by Opiant Pharmaceuticals, which was acquired by Indivior in 2023. And the modeling and simulation activities described by Lafont et al. In their 2024 publications were funded by Indivior. I supported the modeling and simulation activities described by Lafont et al. In collaboration with Simulations Plus and received financial support as a consultant. So opioid-related overdose deaths have been rising in the United States since 1999. And the number of deaths due to opioids was 10 times higher in 2022 relative to 1999. And out of the approximately 108,000 drug-related deaths in 2022, approximately 76% of these were due to opioids. And as you can see in the figure, which is depicting the 12-month ending provisional number of drug overdose deaths by drug or drug class, you can see that in 2024, greater than 90% of opioid-related deaths were due to synthetic opioids such as fentanyl. And you can see that in the black curve, which is depicting the opioid-related deaths, whereas the gold is depicting the deaths due to synthetic opioids, excluding methadone. So there's two available opioid reversal agents in the market, naloxone and nalmefene. So naloxone is an antagonist at the mu-opioid receptor. It has a half-life of 30 to 120 minutes. That's metabolized largely by the liver. And it can be administered intranasally, intravenously, intramuscularly, and subcutaneously. Nalmefene is an antagonist of the mu-opioid receptor and the delta opioid receptor. And it has a longer half-life of 8 to 11 hours. And is also metabolized largely by the liver and can be administered via the same routes of administration. So FDA's translational model integrates the opioid and opioid reversal agent pharmacokinetics, as well as drug binding to the mu-opioid receptor, the pharmacodynamic effects of these drugs, and physiological variables that account for ventilatory control, blood flow control, gas exchange, and metabolism, as depicted in this figure from the 2022 Man et al. Publication. So FDA's translational model, there's two phases to the modeling and simulation activities. In the first phase was a model calibration phase. In this model calibration phase, PK, PD, physiological, and receptor binding submodels were built using existing data, as well as data that were generated through in vitro binding experiments. PK submodels were included for various opioids and the opioid reversal agent naloxone administered via intramuscular administration. And the physiology submodel within this framework was based on two publications by Racino et al. Published in 2001. And this physiology submodel was calibrated further based on ventilatory response data under various conditions. The PD model was calibrated using clinical data from chronic opioid users as well as opioid naive study participants. And in this work, a virtual population of 2,000 patients was developed by combining distributions of both pharmacokinetic as well as receptor binding parameters. In the model validation phase, the ordinary differential equations as well as the parameter estimates were quote-unquote frozen, and additional in vitro binding data, animal studies of cardiac arrest, as well as human ventilation studies with fentanyl, and remifentanil were leveraged for model validation. To perform model validation, model simulations were performed and compared to observed validation data that were not used during the model calibration phase. Once the model was deemed validated, opioid overdose simulations were performed. In these opioid overdose simulations, both medium and high intravenous doses of fentanyl and carfentanil were simulated. For fentanyl, these doses were 1.63 and 2.97 milligrams. And for carfentanil, they were 0.012 and 0.022 milligrams. And these doses were selected based on information from postmortem data. For each dose, virtual typical patients and virtual populations were simulated, and the physiological variables as well as the PK/PD outputs were evaluated. In these simulations, after the minute ventilation drops below 40% of the baseline value, there's a 1-minute delay was assumed for the administration of intramuscular naloxone. So shown here are some of the results from this 2022 Man et al. Publication. So in panel A, you can see results for a simulation of a typical patient, whereas in panel B, you see the results for cardiac arrest for a patient population. So let's walk through these results. You can see that, again, there's 2 doses of fentanyl that were simulated, 1.63 milligrams and 2.97 milligrams, and there was 2 doses of carfentanil that were simulated, 0.012 and 0.022 milligrams. If we focus on this first column and panel A, we can look at the results for some of these physiological variables that were simulated. You'll notice that in this column, we have 3 curves. The red curve that denotes the scenario where we have no naloxone administered. The blue curve denotes where we have the 2 milligram per 0.4 ml intramuscular naloxone formulation, which is a more concentrated intramuscular formulation that's no longer on the market. And there is a 2 milligram per 2 ml intramuscular naloxone formulation depicted by the green curve. If we focus on these first two rows, you notice that what's being depicted is minute ventilation as a function of time in this first row, and arterial oxygen partial pressure as a function of time in this second row. And what's being depicted is a 15-minute simulation window. You can see that if you focus in on the red curve, you can see that both for minute ventilation as well as for arterial oxygen partial pressure, after the opioid is stimulated or introduced into the simulation, in this case fentanyl 1.63 milligrams, you have a drop in the minute ventilation as well as a drop in the arterial oxygen partial pressure. And that red curve eventually culminates in a red cross, which is depicting cardiac arrest in the absence of naloxone intervention. In contrast, if you look at the red curve, the blue curve and the green curve, you can see that there is a rebounding in the minute ventilation as well as the arterial oxygen partial pressure. And you can see that rebounding is occurring quicker with the blue curve, which again is a more concentrated form of the intramuscular naloxone, as compared to the green curve, which is a less concentrated formulation. And this difference between the blue and the green curve potentially may be due to differences in the absorption between these two formulations. If we now focus on cardiac output, which is again this third row here, and again what's being depicted as cardiac output as a function of time in this third row, you can see it across all three scenarios. There is an increase in the cardiac output, which is a compensatory mechanism. And with the red curve, cardiac output eventually around 7.5 minutes begins to drop. And again, that culminates in this red cross, which is depicting cardiac arrest. In contrast with the blue curve and the green curve, you see that there is, after that compensatory increase in cardiac output, there is a return to near baseline values of cardiac output, and that return to baseline is occurring quicker with the blue curve as compared to the green curve. If we now look at one of the doses of carfentanil, the medium dose of 0.012 milligrams, you can see that we have generally similar trends in that the red curve, again, you have a decrease in the arterial oxygen partial pressure, as well as the minute ventilation as a function of time. And in the absence of naloxone intervention, that's culminating in cardiac arrest with the red cross. In contrast, with the blue curve and the green curve, you have a rebounding in these two physiological variables, and that rebounding is occurring quicker with the more concentrated intramuscular formulation as compared to the green, less concentrated intramuscular formulation. If you compare the curves between carfentanil and the ones we just observed with fentanyl, you'll notice that this increase in minute ventilation and arterial oxygen partial pressure after administration of naloxone is not as quick with carfentanil as with fentanyl, and that's because of a slower dissociation with carfentanil as compared to fentanyl. And again, if we look at cardiac output as a function of time, we see a similar trend in that there's this compensatory increase in cardiac output across all these scenarios. And again, there's this decrease in cardiac output in the absence of naloxone intramuscular formulation administration, culminating in cardiac arrest, whereas there's a return to near baseline values of cardiac output with the blue and the green curves, and again, occurring quicker with the blue curves. If we now focus on panel B, which is depicting using bar charts, the percent of virtual patients experiencing cardiac arrest in a simulated virtual population. And again, we're looking at these three same scenarios again across these two different fentanyl doses as well as two different carfentanil doses. If we focus on the red bar charts, what you can see is across all these scenarios, in the absence of intramuscular naloxone administration, cardiac arrest percentages are rather high and vary between 50 up close to upwards of 90% in the absence of naloxone. When naloxone is administered, you see that there's a significant decrease in the percent of virtual patients experiencing cardiac arrest. That decrease is greater with the more concentrated form of intramuscular naloxone as compared to the less concentrated 2 milligram per 2 ml across these different scenarios. If you look at cardiac arrest as a function of dose of fentanyl as well as carfentanil, as you would anticipate, as the carfentanil dose goes up, as well as the carfentanil dose goes up, you see increases in simulated patients experiencing cardiac arrest. And if you compare fentanyl to carfentanil, you can see that generally cardiac arrest percentages are higher with carfentanil as compared to fentanyl. So this FDA translational model was further applied in this 2024 publication by Strauss et al. In this publication, intranasal naloxone was integrated into the model framework, and also explored within this publication are repeat doses of intranasal naloxone, which is what's being depicted here in this blue rectangle portion of the figure. You can see that there's 2 doses of fentanyl again being simulated. In gray, we have fentanyl 1.63 milligrams, and in blue, we have fentanyl 2.97 milligrams, both intravenously administered. You can see here that there's either 1 or 2 doses being compared at the 0-minute time point, and then there's also additional doses that are being evaluated at both 2.5, 5, and 7.5 minutes. And if you compare the results, what you can see is that the simulated cardiac arrest percentage decreased as a function of the initial dose, so 1 versus 2 doses, but not with repeat dosing every 2.5 minutes. And that's what you're seeing here between these 3 points versus these 2 here, which are depicting the 2 initial doses. And you see a similar trend with both the 1.63 as well as the 2.97 milligram intravenous fentanyl doses. So in Lafont et al. 2024 publication, the authors leveraged the Mann et al. Model to compare the simulated cardiac arrest percentages following intranasal administration of naloxone or nalmefene. In terms of the integration of new components, pharmacokinetic parameter estimates for intranasal naloxone, intranasal nalmefene from a population PK analysis were integrated into this model framework using ordinary differential equations. And then the nalmefene binding parameters were obtained using a scaling approach, such that binding parameters reported by Cassell et al. In 2005 were leveraged. More specifically, the ratios of nalmefene K-on to naloxone K-on and nalmefene K-off to naloxone K-off were multiplied by the naloxone K-on and K-off estimates used in the model, in the Mann et al. Model framework. Additional model validation was performed. Specifically, the data from Ellison et al. 2024 were used to validate these new model components. Briefly, in the Ellison et al. Study, healthy volunteers breathed a hypercapnic gas mixture. 10 minutes later, remifentanil was administered as an IV bolus dose, followed by an IV infusion. And 15 minutes after the start of the infusion, study participants received 3 milligrams IN nalmefene hydrochloride or 4 milligrams IN naloxone hydrochloride. Remifentanil pharmacokinetic parameters and binding parameters were reported by Eleveld et al. And Mann et al. Respectively. Again, similar to the Mann et al. Publications, once this model validation was performed, opioid overdose simulations were performed. And in these opioid overdose simulations, model simulations were performed to evaluate the simulated cardiac arrest percentage in virtual chronic opioid users and virtual opioid naive individuals, both in the presence and absence of intranasal opioid reversal agents. So depicted here are the results from this additional model validation using the data from Ellison et al. What you can see in both panels A and B, what's being depicted is minute ventilation as a function of time. And in panel A, what's being depicted is a scenario where study participants receive intranasal nalmefene, and in the panel B is intranasal naloxone. And what you can see is that the simulated minute ventilation in a typical virtual opioid naive individual, which is depicted by the black line, was in good agreement with the mean observed data, which is depicted in the orange line, as well as the individual level gray circles and observed data. Again, these data come from the publication by Ellison et al. So this supports that the integration of these new components of the model describe these data well. So again, these opioid overdose simulations were performed once these new components were integrated into the model framework. And again, we're looking at results here that are similar to the prior results that we reviewed from the Man et al. Publication. Again, we have 4 different scenarios being compared, the same 2 fentanyl doses and the same 2 carfentanil doses. And again, like before, if we focus in on this these first column, which is the fentanyl 1.63 milligram dose, again, you can see that for the first 2 rows, which are minute ventilation as a function of time and arterial oxygen partial pressure as a function of time, the red curve, which again depicts the scenario where no opioid reversal agent is introduced into the simulation, you can see that you have a decrease in minute ventilation and a decrease in arterial oxygen partial pressure, ultimately culminating in this red cross, which depicts cardiac arrest. In contrast, you see with the blue curves as well as the green curves, you see a rebound in these physiological variables. In this case, the blue curve depicts 3 milligrams intranasal nalmefene, and the green curve depicts 4 milligrams intranasal naloxone. And you can see if you compare the blue and the green curves that you have a rebounding again in these physiological variables for both these curves. But if you compare the blue and the green curves, it appears that this rebounding is occurring quicker with IN nalmefene as compared to IN naloxone in this simulation. Again, if we look at cardiac output across all these scenarios, you have this compensatory increase in cardiac output across all these scenarios. And then you have this decrease in cardiac output and culminating in cardiac arrest in the absence of administration of any opioid reversal agent. Whereas you have this return to baseline values of cardiac output when either naloxone and nalmefene are administered, but you can see that the blue curve, the return to baseline is occurring quicker for IN nalmefene in the simulation compared to IN naloxone. Again, if we look at if we focus in on, let's say, on the medium dose of carfentanil, 0.012 milligrams, and we look at these physiological variables again, we see that there is this decrease in minute ventilation, arterial oxygen partial pressure, culminating in cardiac arrest in the absence of an opioid reversal agent. But we have a rebounding in minute ventilation, and we have a rebounding in arterial oxygen partial pressure when either nalmefene or naloxone administered, and the rebounding is occurring quicker with the blue curve, which is intranasal nalmefene. Again, if we look at cardiac output, a similar trend, such that cardiac output eventually increases across all scenarios, ultimately culminates in cardiac arrest with a red curve, and then a return to baseline with the blue and green curves, and the return to baseline is occurring quicker with the blue curve, which is IN nalmefene. Again, if we now look at the percent cardiac arrest in a virtual population of individuals, again, you can see that we see a similar trend in the absence of any intervention. Cardiac arrest percentages are high, varying from 50 upwards, close to 90%, whereas with administration of both naloxone and nalmefene, there's a decrease in the cardiac arrest percentage. And across these different opioid and opioid doses, you can see that there is a greater reduction in the cardiac arrest percentage with nalmefene, depicted in the blue chart, in the blue bars, as compared to the green bars, which is intranasal naloxone. So shown in this figure are similar results. These are cardiac arrest percentage results, but now we're looking at here in terms of tabular format. If we focus in on the intranasal simulations, and we compare the 4 milligram naloxone intranasal dose, single dose, as compared to a 3 milligram nalmefene dose, and you compare the cardiac arrest percentages across these different fentanyl doses as well as carfentanil doses, you can see that there's lower cardiac arrest percentages with nalmefene as compared to naloxone. And you can notice that multiple doses were also of naloxone as well as nalmefene were also evaluated. These doses in these simulations were administered simultaneously, And in these simulations, they were administered simultaneously, based on the publication by Strauss et al., suggesting that initial doses, increases in those initial doses were beneficial, whereas doses further out at 2.5, 5, and 7.5 minutes were not beneficial. So in these cases, these doses are being, additional doses are being simulated and administered simultaneously in the simulation. And you can see that you have to give 4 doses of 4 milligram naloxone simultaneously at the same time in order to obtain simulated cardiac arrest percentages that approach those simulated values for a single dose of intranasal nalmefene 3 milligrams. And the results here that are being depicted are for chronic, virtual chronic opioid users. In this publication, also simulated were virtual opioid naive subjects. And again, if you look at 4 milligrams IN naloxone versus a single dose of 3 milligrams IN nalmefene, in these virtual opioid naive subjects, you see similar trends where the simulated cardiac arrest percentages were higher with the intranasal nalmefene as compared to the single dose intranasal naloxone. And again, it's in the scenario where you get 4 simultaneous doses of 4 milligrams of IN naloxone, where these simulated cardiac arrest percentages approach those for a single dose of intranasal nalmefene. If you compare these simulated cardiac arrest percentages with the values reported in the prior slide, you can see that, as you would expect, the simulated percentages in virtual opioid naive subjects were generally higher than those in virtual chronic opioid users. So to summarize, the FDA's translational model described by Mann et al. And Strauss et al. Provides a mechanistic framework to evaluate a plethora of physiological effects and consequences of opioid overdose and the ability of opioid reversal agents to reverse opioid-induced respiratory depression. Lafont et al. Leveraged the FDA's translational model incorporating opioid reversal agent intranasal nalmefene, replicating the intranasal naloxone data reported by Strauss et al. Using a different PK data set, and additional validation simulations were performed. Simulations using the validated translational model of opioid overdose demonstrate that a single dose of 3 milligrams IN nalmefene produced clinically meaningful reductions in the incidence of cardiac arrest compared to 4 milligrams of IN naloxone following a synthetic opioid overdose. And simultaneous administration of 4 doses of IN naloxone were needed to approach a simulated incidence of cardiac arrest obtained with a single 3 milligram dose of IN nalmefene. And with that, I thank you for your attention, and I'll turn it over to Dr. Delapine. Thank you.
Thank you so very much, Dr. Gonzalez. And Jasmine is going to launch a question for people to answer, a poll question. So if you would answer, you would have a QR code to do that. And that's for the whole audience.
Yeah, so you'll see a poll question pop up. It's just asking if you're interested, if somebody, for someone to follow up with you, and I'll give everybody a couple seconds to reply there.
And also a reminder that if you want to add your questions, there's a Q&A button for you to use, and type your questions in there. And we will start soon.
All right, I went ahead and ended.
Very good, thank you. So while we get some more, let me ask the panel then first, what are for you the most important takeaways from reversing an overdose from what we've seen from the model? Dr. Gold, do you want to start?
Sure. So like for me, a model's a model, and congratulations on doing this work, Dr. Gonzalez. I'm of course biased, having worked for so many years with the College of Pharmacy at the University of Florida, and the work is outstanding. So the idea of having models is very important and useful to me. My own thinking about this is, of course, what's the human condition, and how can we relate that to the human condition, and how can we relate it to the choices that emergency physicians, Dr. Boyer and his colleagues have to make about what agent to use and when. So the model's been very helpful in trying to convince us that it is better to load up the initial doses of naloxone rather than titrate it in over time. However, my thinking as a neuroscientist is always, well, oxygenation is one measure and cardiac arrest is another measure. All of these overdoses tend to occur in people who are chronic recidivists, while death occurs to people who've taken a fentanyl containing bootleg tablet. The majority of the cases tend to be people who are chronic relapsing and use opioids. We have no idea as clinicians, by the way, how many times they've overdosed. We don't have any idea how many times they've been reversed. We don't really have any idea how severe the overdoses have been. And so only recently have my colleagues begun to look at the consequences of anoxia, which is can be seen residually in the brain. So MRI studies at Penn and with help of NIDA have shown that people who've had overdoses have, in addition to the chance of death, even when they are rescued, it appears that their residual neurological deficits and evidence of brain changes that appear likely to be long-lasting, if not permanent. I reviewed this in one of my Psychology Today posts and heard back and forth from experts at NIDA. The outcome measure of death is one thing, and the outcome measure of cardiac arrest is another, but the overwhelming majority of people who've had an overdose and a reversal, they kind of assume that the consequences of reversal would be complete and they wouldn't be left with neurological deficits. The data here in the model here suggests that the oxygenation delta between naloxone and nalmefene creates a scenario where it would be possible to be rescued and have CNS damage, whereas with one versus the other. And I just wonder, Dr. Gonzalez, whether there's a way to look at that and model the brain cell challenges in this hypoxic environment and the effect of time, the time delta between the doses in reversing hypoxia, independent of the fact that so many of our patients have had repeat events and that the damage then becomes cumulative. But everyone's goal is to preserve life, but in addition, we need to preserve brain function.
Dr. Dahan, would you like to comment on that, or Dr. Gonzalez, would you? I think the modeling can always go on, right? As long as we have the appropriate data to inform the model. Sorry, this keeps popping.
Can I comment on the model?
Yes, please.
Or ask questions regarding the model? First of all, it's a great attempt to mimic an opioid overdose. In fact, there are three conditions in which people overdose. That's the ones, people that abuse specifically opioids, the ones that are inadvertently overdosed by taking other drugs, and in case of a pharma terrorism attack in which in the environment opioids are released. If we look at the reversal strategy that has been applied in this model, but also in the man model, I was part of that study as well. I find it a little bit too optimistic. How long does it take before you reverse someone that has overdosed? it's not 1 minute after 40% of respiratory depression, far from it. It's at least 7 to 10 minutes after the actual overdose. And these individuals have been apneic without breathing, often with very low cardiac outputs, high CO2s for many, many minutes. And I agree with the previous speaker. The likelihood of brain damage is extremely high. And we hear very little about that. I wrote an article on that, and that I'm so surprised by that. So I know, because I was a couple of weeks ago at the BARDA meeting on medical countermeasures for pharma terrorism. I know the data is there. I would love to see what happens when you reverse with nalmefene after 20 minutes, after 30 minutes, very, very low cardiac outputs. Because the increase in cardiac output is a compensatory mechanism because of the high CO2, evidently. But at one point, at these high CO2s and low O2s, you get a reduction in the heart rate, and you get high probability of cardiac dysrhythmia because of the high CO2. So that's my first request. If we could see some later data, that would be very much of interest. And I know the data is there. The second issue I have are the pharmacokinetics. I myself recently performed a study in which we gave multiple doses of naloxone to both healthy volunteers and individuals with an OUD. And the issue is a little bit the fact that there is saturation and uptake intranasally. So by giving 4 milligrams naloxone, we observed that the increase in the plasma concentration is very minimal. In fact, after 2 doses, the increase of naloxone is very limited to 3, 4. You don't even have to give more than 2 doses. Having said that, there is another option of administration of naloxone. That's the intramuscular pathway. And when you do that, you do see much higher concentrations of naloxone, much more rapid reversals, even with very low cardiac output levels. So my main question is, what happens when you delay your reversal strategy? Maybe Daniel can, maybe you have the slides.
Yeah, certainly. I mean, the model can be used to evaluate delays as well in the opioid reversal agents, as well as brain oxygenation as a function of time. And those simulations are one that are also being considered and performed.
Yes, Dr. Lee, would you like to comment?
Yes, sure. Can you hear me?
Yes.
Yes. First of all, I'm glad that the model we have developed were used in this study. But first, I would like to caution against making any general statement about nalmefene versus naloxone, because we all know that it depends on according to our recent research, it all depends on the absorption rate for the same formulation. Depending on what absorption enhancer you put into the formulation, the same formulation can show very different performance. So I think when we make statement about comparison, we should consider the PK, the exact PK for each individual formulations, rather than saying nalmefene versus naloxone. But that being said, when we reviewed this nalmefene intranasal 3 milligram product, we did apply this model in the review as well. The results we obtained were similar or qualitatively similar to what was presented today. I mean, there are some differences quantitatively, probably because we used more data during our review. For example, the sponsor measured not only medium ventilation, but also carbon dioxide time course in the study. They also performed a part 1 preliminary study where they measured the so-called mask effect, meaning just by removing mask without giving any naloxone or nalmefene, there was some fluctuations in medium ventilation. So in our review, we did put those into consideration, but the final results, we also show that this particular formulation of nalmefene was predicted to have less cardiac arrest compared to the 4 milligram intranasal naloxone, but the difference was not as huge as what was shown today. I guess the lessons we learned was that because this model has so many parameters, when we try to apply it, we should use as much data from the same study as possible to fully constrain the parameters so that the simulation is as faithful to the real condition as possible. And also, I would like to comment on the question raised by both Mark and Albert. Yes, brain damage was right now. We did have this brain hypoxia time of the endpoint for the model, but we did not. So we could predict the time it takes for the brain hypoxia to be reversed, but we did not. So far for the model, we do not have the component to predict the long-term chronic damage to the brain due to repeated hypoxia. That's indeed something we are considering. And Albert, you said there's some data out there, but if we can get more data about those chronic brain damage, I think that will really help us to understand the consequence of this overdose.
That is a great point, and I believe there's more data being collected as we speak on these endpoints. We did not model brain damage at this point yet, but like I said, everything is possible if we have the correct data. Dr. Boyer, do we have you as well? Would you like to comment?
Yeah, hold on. I've got real difficulties with the internet at work. I mean, if I really liked the comment that it's not about life, it's about brain function. And, I know that, there's an interest in getting people to use, different products for reversal of opioid overdose. But, you're right. It's not life, it's brain function. And I think that's why, when I've talked to people about these studies, the universal complaint has been, who cares about cardiac arrest? Like I don't care about cardiac arrest as an endpoint. I mean, I get that it's a good hard endpoint. It produces a good hard dichotomous model or dichotomous finding, but that's still going to be a point that people are going to have to address at some point. At least with emergency physicians and paramedics, if you want people to use this. I think the interesting thing here that I wasn't aware of is that the intranasal uptake of naloxone appears to be saturable, and a lot of people have switched over to intranasal naloxone just because they don't have to deal with sharps, like in a fraught clinical environment. I think demonstrating to people that the process is saturable, and then you have like upper limits in the amount of naloxone that you can deliver following intranasal administration is, I think, going to be important, particularly since a common comment from a lot of folks is, who cares? I can just give more naloxone. I think toxicologists inherently understand saturable kinetics. You switch from first order to zero order activity, but that may be a bit beyond what most emergency physicians can wrap their head around.
Thank you. We have another question online asking if it can be concluded from Dr. Oliver Grunman, if it can be concluded that nalmefene may be a better intranasal reversal agent than naloxone, and also if you can comment on the half-lives of naloxone versus nalmefene. So I'll direct the first part of the question to our clinical experts, and then the second one to Dr. Gonzalez.
Well, I think what Dr. Lee said, and I think the clinical people would tend to support that, is there are limits to models, and we like to do things in real life. Of course, it would be great to be able to do a direct prospective comparison and look at endpoints that really matter to us, like residual brain function, like brain hypoxia, like and if you take a cardiac measure that as well. It's an interesting question because part of the pushback, I think Dr. Boyer had one pushback, but another is naloxone has become much more than just a reversal agent. It is part of the health delivery system, and the investments that have been made in terms of stocking and dollars and inventory makes it somewhat different to consider. So I played with the idea for a while of well, there's all this naloxone out there, and people are attached to it, and it's in their inventory as well, and it's difficult for them to change. On the other hand, data always is a compelling way to change medical behavior. And I do wonder whether the agents might be used at the same time, whether they might be a direct comparison study that's done that is convincing. But for right now, you have so much, if you look at it like the interests in naloxone are much greater than the current data because of the investment of time, energy, and capital.
I'd actually say one thing, this is Ed Boyer again. I'd actually say one thing a little bit different. We know data doesn't change behavior in healthcare settings. It's actually academic detailing. So trinkets change data. Or trinkets change.
I could have the Boyer reversal agent, but we luckily in addiction medicine we don't have the similar kind of. It's a very interesting question, but again, I think naloxone. I mean, I'm probably the oldest here on the panel, and I remember giving naloxone in the Yale emergency room in 1975. I remember provoking opioid withdrawal and figuring out with colleagues that if you gave repeated doses, each subsequent dose of antagonist produced less withdrawal, and you could pretty much switch someone to naltrexone while they were sitting there in the emergency room, even though their initial discomfort was great. So I haven't found it problematic that there was a risk of extended withdrawal distress. And I think that brain data, once it does exist, can motivate a change in prescribing. And I'd be optimistic about that and see what Ed thinks.
Parag, can I add something
Yes.
quickly, even though it's not direct to me? I
Please.
think, like I mentioned earlier, even with all the limitations of the model, and even when we only use cardiac arrest as the endpoint, when we do comparison between these two formulations, intranasal nalmefene 3 mg versus intranasal naloxone 4 mg, I think the best thing we can say that they are comparable, because when we apply the model to the review using more clinical data, the difference between the two formulations were not that significant. Two is that I think the Lafont paper also limited that the PK in the PK/PD study is different from a quote-unquote pure PK study. So even the same formulation, even by picking different PK study, you may get different PK profile, different absorption rate that will change your result dramatically. So in the end, we may see a mixed results. So I think probably the best thing we can say for now is that the model predicts that these two are comparable.
Dr. Boyer?
Yeah, yeah. I mean, I think the the stream that we have to swim up against is first, familiarity with naloxone. People just get it. Like we've got decades of experience in using it, and like it's understood by everybody from paramedics, by like every arc of treatment, like from paramedics to the physicians to the nurses, everybody gets it. It's easy. It's also forgiving. Like if you give an overdose, it doesn't hurt anybody. If you send somebody into withdrawal, they're unhappy and you get cursed out a lot. There may be some cleaning up to do, but like at the end of the day, you don't have to worry about the patient dying. That's pretty uncommon in the emergency medicine world. That said, the question is, how do you change behavior? And there was a guy at Brigham and Women's Hospital back in the 1980s who said, I want to change clinicians' behavior. And he came up with the entire field of academic detailing that got co-opted by the pharmaceutical community. Getting somebody to change things is not a mystery of how to do it. It's just how to do it with a medication that everybody recognizes is inexpensive and widely available, that everybody understands, that's very forgiving. Also in the context of the Sunshine Act, how much you can actually have access to physicians to try and get them to change their prescribing. That's a business decision, or that's a marketing and business workup, not necessarily a medical one. But like at some point, those two features are going to intersect, and you are going to have to persuade somebody with 30, up to potentially 30, 35 years of experience giving a medication to do something brand spanking new. That's hard.
Yeah, that's a really great point. Dr. Lee, when you said that they're comparable, is that comparing 4 doses of naloxone to 1 dose of nalmefene?
No, just 1 dose. When in our simulation, we also saw a slightly advantage of the nalmefene formulation, but not as significant as the results shown. And also, like the PK can be different in different PK studies, so it's very hard to make a general comparison statement at this point. That's my feeling.
Thanks.
Thank you, everybody. We're 1 minute from the end of our webinar. Thank you so very much for your participation. Jasmine, do you have any closing remarks?
Yeah, I just want to say thank you to all our presenters. If you had a question that we weren't able to answer, or maybe will come up, someone from our BD team will reach out to you. We invite you to visit our website to learn more about our capabilities. You or your colleagues may also be interested in our upcoming webinar. The title is DDI Risk Assessment to Inform Your Label, How to Optimize Your Program's Timelines and Budget for PBPK Modeling. And then you can find links in the chat to that. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you again, everyone.
Thank you, everybody, and Happy New Year again.